Welcome everyone to the Funga Speaker Series. I'm Colin, I'm the founder and CEO of Funga. At Funga, we reintroduce fungal biodiversity to forests to accelerate tree growth and carbon removal as a natural climate solution. And we run this seminar series to try and bring in new ideas from the outside of our organization, as well as give a platform for science in our space that we think is exceptional. And so with that, it's my privilege to introduce Dr. Jennifer Bhatnagar, who's an associate professor of biology at Boston University. I actually did my postdoc research in her lab, and it's awesome to have the chance to bring her in here. Dr. Bhatnagar has a background in chemistry and has a reputation for being one of the first to bring cutting-edge chemical molecular tools to the study of mycorrhizal symbiosis, which is pretty awesome. And today, I think she's going to tell us about some super new work she's done on the restoration of microbial biodiversity in urban forests and on the use of microbial biodiversity in biotech more broadly. And with that, I'll hand it over to you, and hopefully the screen share works.
Great. Thanks, Colin. Yeah, so hi, everybody. Let's give this a shot. Let me see if I can share my screen. Host disabled participant screen sharing.
I'm going to fix that right now. Cool.
Oh, there we go. Got it. Okay, so thanks for coming, everybody. As Colin mentioned, I'm quite new to the restoration and ecosystem environmental engineering space. All of my work for the last 32 years has been fundamental science about plants and microorganisms in forests. So my background is pretty squarely in forests, but I moved into restoration because of the students, my graduate students really, who for whom I think fundamental science is no longer enough. I think they really want to see impacts in their lifetime of the work that they're doing, the scientific research they're doing. So that's why I'm here. Also inspired by Colin, who has been a real pioneer in this space and made me feel like I could do it maybe. So I asked ChatGPT to make up a name for the stuff that we're doing, and it came up with microbiome ecotechnology. I have very mixed feelings about that. I wonder what you all think. Ecotechnology has a history, like as a term. I didn't know it existed, but it's mostly, I think, been referred to for things like geoengineering and stuff, like physical engineering of the environment. But I'm gonna take it and put it with microbes. So this work I'm going to talk about, it was really driven by my current postdoc, Chikai, and my current graduate student. And it's motivated by this observation that this was made in 2019 for the first time. This observation that city trees die twice as fast as rural trees. This happens in Boston, where we live. It also happens other places around the world. And we don't know why. But one idea is that maybe this is related to the microbiome of these trees. So here's a question for all of you. What kinds of microorganisms do you think might be able to survive in a city? Maybe even specifically fungi that you know of. You can put your ideas in the chat. Stress tolerators, okay, that's one idea. Ones that are surviving in the city, okay. Other ideas, pathogens. Okay. So there's this idea that cities are hard. Cities are hard living, and they are because cities are made for people. They're not made for anything else, right? They're not made for any other organisms. The things that can live there are weird. Um, Cities have not been studied nearly as much as beautiful, pristine rural places. And that's for a variety of reasons, but people are just starting to study them now. And what they are finding is that, yeah, there's some evidence that pathogens might be ticking up in cities. This paper came out in 2021. It's the first one I could find on this, and actually the only one since then, that shows that there are a lot of pathogens living on the leaves of oak trees in cities. So we came up with this hypothesis that pathogens flourish when symbionts go down. There's very little evidence in the literature to suggest this is the case, but it's funny, I've noticed a lot of review papers that have come out in the last years that talk about systemic responses of plants to mycorrhizal inoculations. So inoculations with ectomycorrhizal fungi and arbuscular. And there is this idea that somehow inoculation of plants with these fungi can allow plants to resist all sorts of stressors throughout the entire organism of the plant. So we decided to see if urbanization affects this and forest fragmentation in a gradient study of oak trees in Massachusetts. And so I'm just going to very briefly cover a couple of things we found here before I get into the restoration part. I think you can guess what this story is going to be. So in these little forest plots, so these are forests, right? These are not single trees yet. I will talk about single trees in a minute, but forests, we have this urbanization gradient going from Western Mass over to the city of Boston, which is the star right here. And at each of these spots, we looked at the microbiome of soils actually underneath oak trees, right at the forest edge, like next to a roadside or an agricultural field, into the forest interior. And this is a collaborative project with some other PIs, Pam Templer and Lucy Huteri here at BU. And so Chakai did a lot of this work, and what she found is what you might expect, which is that ectomycorrhizal fungi don't do super well here, but it's very specific. Mycorrhizal fungi that live on oak roots decline at forest edges. And you can see that over here on the left. So for both rural forests and urban forests, they go way down at the forest edge. Even though there are a bunch of roots there and there are a bunch of oak trees growing, the biomass of oaks doesn't change across this gradient. But we noticed another interesting thing. Urban forests in this part of the world have a lot of ectomycorrhizal fungi in them in the soils. We think this is because, for some reason, urban forests have a lot more oak trees than rural forests, which are more mixed in terms of tree species composition. Nevertheless, the fungi, the ectomycorrhizal fungi in these urban forests do not colonize the oak trees as much as they do in a rural forest. So all these mycorrhizal fungi are in soils, but they're not doing their usual jobs. They're not colonizing oaks. And instead, what we see actually is that when the ectos go down at the forest edge, pathogens are ticking up, plant pathogens and animal pathogens. Now, we've recently taken this, that was published last year, that information, but now we've moved to even more severe urban areas, street trees. And this is work from my PhD student, Katie Atherton, found that street trees have even fewer ectomycorrhizal fungi than these forests, these urban and rural forests. And this is pretty strongly related to nitrates in soil, but also pH and temperature. Cities are hot below ground, really hot. They're about 10 degrees C hotter than rural forests in this region. They're extremely hot in the summer due to the urban heat island effect. And ectos don't like it. Interestingly, the ectomycorrhizal fungi are almost completely replaced by wood saprotrophs, and I think it's because the trees are dying and the saps are kind of in between pathogens and saprotrophs, and they're taking advantage of these super duper stressed out trees. Okay, one last piece of information before I get into the restoration, which is that things are weird above ground too in cities. So what I'm showing you here is some data from the same trees at an increasing distance from the city out to Western Mass. And you can see that, oh, I didn't put a legend on here, sorry. The blue line here, these are sooty molds. So this is a plant pathogen, a very specific type of fungal pathogen that is accumulating on leaves in the city. They are actually only present when there are aphids because they eat the honeydew that the aphids create when they eat the leaves of the oak tree. So we're getting a lot of pathogen damage of the trees, and these molds are growing as a result. We also have a decline in endophytes and epiphytes on the surface of the leaves. And you can see, the epiphytes are really going down with nitrogen deposition, whereas the sooty molds love it. They're really responding positively to both temperature and nitrogen deposition on the leaves. And these endophytes seem to be tracking different types of nitrous oxide and nitrogen dioxide deposition as well. Okay, so the microbiome of trees is super weird in the city. We're seeing a loss of mutualists, your classic mutualists like your mycorrhizal fungi and roots, but also potential mutualists like foliar endophytes and epiphytes on leaves, and this rise in pathogens. It's like a horror movie, like really like tons of pathogens on these guys on the below ground and above ground. And so what do we do? Right now we're trying to figure out if this is related to tree death. Looks like it probably is on some level. So can we change that? Can we reverse it? Especially given that these trees don't just show up. They don't just like start growing. We are planting them in cities, and cities are planting them all over the world. And they're spending millions and millions and millions of dollars to plant trees. And by the way, most of the millions of dollars is actually spent on maintaining trees, trying to prevent them from dying, and then replanting them when they do inevitably die. So I want to talk about 3 things we're trying to see if they work. I'm going to tell you up front, we don't know if they're going to work yet. But this is based on the forestry literature. People go out and inoculate trees in rural forests. And, I think a lot of Colin's work at Funga has inspired this as well. So I'm going to talk about 3 ideas. One is inoculating with mycorrhizal spores. Another idea is inoculating roots of these trees with whole soil, pieces of soil, which I know Mia has done a lot of. And the other is leveraging this kind of new idea of a nurse plant to bulk up the microbial biomass, like the beneficial microbial biomass in soil that a new tree can tap into. Any questions at this point? Okay, I'll keep rolling then. So, spores. I thought about spores, and the reason I thought about spores is because there's a lot of evidence from forest restoration work that squirting a bunch of spores onto, little seedlings and saplings can make the plant grow better, grow faster, access nutrients in the soil better. Turns out the city of Boston decided they're just going to do it. And what they're doing is they're using a commercial kit. So all new trees planted in the city of Boston as of last year, 2023, are being planted with a mycorrhizal spore mix. This is the mix, and at first I was like, well, this is dumb, but for various reasons. But then I looked at this list of taxa, and I'll tell you, this is exactly the list I would have picked if I was designing it myself. Now, where do these spores come from? I don't know. And that's really problematic, I think. And we'll get to that later. But this is an interesting mix of both ectomycorrhizal spores and arbuscular mycorrhizal, although you'll see the AMF are not diverse. They're all in the genus Glomus for whatever reason. Okay, so now we have this sort of natural experiment where we can see if we have a lot of data on tree growth and death for the last 20 years in Boston. And now we can kind of compare that going forward to all of the new trees that are planted, how they're going to live and die. To be honest, I'm just not quite sure what to expect here because, as I said, I would have picked those taxa, at least for ectomycorrhizal fungi, to include in a mix because they're very abundant in cities, actually. They're super stress tolerant. They can often confer stress tolerance to trees. But we also have this data here, like on this graph, that show that commercial spore packets are not species specific and they're not really reliable. So this is data from a 2022 paper, folks over in the UK, where I think, oh, I'm sorry, I think it was the UK, in Europe, where they hit a bunch of different plant species with a commercial spore packet of AMF, so arbuscular mycorrhizal spores. And they compared the biomass, the growth of these plants with control, like doing nothing. And you can see the variation. So they did it for plants in Australia and plants in Europe. And sometimes it works super great. And sometimes it totally doesn't work and it's a huge waste of time. So, we're still in the kind of discovery exploratory phase, I think, of working with commercial spore packets. Often they're dead, sometimes they're not. It's almost impossible for us right now to predict the response. So that's TBD. We'll see. We'll see how spore inoculations go for trees in the city. And I'm curious if you all have experience with spore inoculations from commercial spore packets for trees and what you have found. Okay, next is whole soil inoculations. So whole soil inoculations, we've been thinking about for a while. This is work that Colin has done a lot of while he was with us, but also since then, obviously, we're still doing it in our lab. And what we find is oftentimes when you grow plants from seed in live soil, they just develop these beautiful symbioses with fungi in the roots, and it's the bomb. However, there are issues with this. One issue is one issue is pathogens. We definitely notice that, especially with certain plants, maybe all plants that aren't conifers, they can get eaten up by pathogens in the soil before they establish. Another issue with this, of course, is sourcing the soil. And I get a lot of questions about that from people who want to inoculate with host soils, but just aren't quite sure where to get it from. So a third issue is like, what's the best soil? When you're dealing with plants in the city, will they do best with city soil and microbes that are adapted to the city, kind of like you suggested in the chat? Or do we want this beautiful, rich, rural forest soil that has all the microbes that we think are the best? And so to test that, we designed this project called the Transplant Project, where we're just dumping in all different types of soil into these poor plants and exposing them to these horrible urban conditions to see what sticks. And this is preliminary data. This is actually after 1 year of growth. After 1 year of growth, it actually looks like urban forest soils are the best for these little oaks growing in these terrible conditions. And this sort of jives with what we found in these urban forests where they have a lot of ectomycorrhizal inocula and very, very low pathogen loads. So like, duh, that sort of makes sense. Like find the soils that have the good stuff and not so much of the bad stuff. Unfortunately, you have to go through the trouble often of doing all the sequencing or the detection of these fungi to know. But it's also possible that maybe they're just better adapted to these conditions. So that's ongoing, and we'll see how that shakes out after year 2. But the last thing I want to talk about is nurse plants. So there's this beautiful idea in the world that trees and other plants can help each other live. This is a really nice graphic from Courtney Good, who's a graphic designer at RISD, and studies fungi in graphic design and in landscape design. And maybe if these two plants are sharing mycorrhizae, they help each other. They can one plant that's not doing well or maybe just starting to grow can be assisted by the plant that's connected to the same fungus underground. I don't know. Nobody really knows how extensive that is and if it really helps. So we set up this project out in California. So this is a different ecosystem. This is a very urbanized system that's experiencing extreme wildfire. So there's a lot of interest in replanting out there. We're calling it Sprout Oak, where we're planting these oaks that are threatened and endangered out there due to agriculture and urbanization and now intense wildfire. And we're planting them with and without a nurse plant. We did this weird thing where we planted them with ectomycorrhizal nurse plants where they should be sharing ectomycorrhizal fungi, but we also planted them with nurse plants that have arbuscular mycorrhizal fungi. And this is because there's some evidence that in the very early stages of life, oak trees are arbuscular mycorrhizal and not ecto. And for some reason, people don't like to talk about that or think about it. But we thought we really want to find the best thing for these trees because they're so coveted and people enjoy them so much that why not? So this is ongoing. My PhD student is out there harvesting this project right now. And it is interesting. So far, I think we're completely wrong. It turns out that the oaks just tend to not do very well when the nurse plant is nearby. They're just too slow growing. So what we're doing is testing this again, but out in the world. We're right now, the end of this month, going out to California to plant 45 Miyawaki forests out in Napa Valley, where we're focusing on oaks, but we're co-planting them with nurse plants at a distance with different types of soil inoculum to see if this actually does help them when they're out in this very stressful post-fire environment. And we're measuring all sorts of things on that to see if these treatments, how they impact oak growth, but also just total forest regrowth. And this has been a really interesting project because most of the land in California is privately owned. So these are all on like people's properties that burned, and they want very specific types of plants there. They don't want pines. They don't want any conifer. I know, but they don't. And I was trying to tell them to do Douglas fir, and they like bit my head off. Douglas fir is great because it grows fast. It's like this model species. So it's great for science, but it's bad for people because it lights up like kindling. So this has been a really interesting socio-ecological project. We're like working with people, sending them a plant list. They're like, no, 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 I don't want this one. I want this instead. I want strawberry. I want, sage. I want whatever, right? And so co-designing this with the community has been really fun and really educational for us. So that's it for me. Thanks to you all for listening, and I'm happy to chat about this and take some questions.
Awesome. Thank you so much, Jenny.
Sure.
Feel free to raise your hand and or just unmute yourself and ask a question, or if you prefer, drop a question in the chat and I can read it out. And I'm happy to start with a question while everybody thinks of what they're going to ask you. But first of all, you said your students were coming to you, asking you for applied science. And I was wondering if you could just share any more about that or an anecdote. Like,
Oh,
is that something that wasn't happening 5 or 10 years ago that you feel like is happening now?
Totally. I mean, we still get a lot of students that want to do like fundamental stuff, but I think it's hard to say. Actually, I should go back and do the numbers on it. But I think it also maybe coincided with a change in my interests a little bit. Like that's probably what also happened where I became, especially with the city stuff, because Boss BU, the concrete campus, we're right here. I look outside and there's street trees right there, and they're just the saddest, just the most pathetic. It's heart-wrenching. It's awful. And actually, what happened is, so I had a PhD student, I still have one, Katie, actually, the one who generated all this phenomenal data. She came and she said, I want to rewild urban areas with microbes. And I was like, nope, we're not doing that. We're doing fundamental science. And, she's Katie, so she's a trooper. She went and did her fundamental science, but then the pandemic happened and I was home a lot. I had two babies and I was home all the time with them. And I remember looking outside and there was a playground being rebuilt near my house and they were planting trees. And I would yell out the window. I would like open the window and be like, stop planting, because I knew they were going to die. They would just truck in this urban fill from like who knows where, sandy, loamy, whatever, from Home Depot, just spread it and then pop the trees in. And you know what they would do? What I found out is they would, you probably know this, they sterilize the roots. So they dunk the roots in like antibiotic and an antifungal, and then they plant them, and then they die.
That's
And
wild.
and I thought, God, that is so stupid. And then I started talking to the people who are doing this tree planting and tree management, and they said, yeah, but A, we've got to get rid of the pathogens. We have a huge root pathogen problem in the city. That's the only way we know to get rid of them. And B, we don't know what else to do. Tell us what to do, scientists. Because like we're trying our best and we're not microbial scientists. Like we don't know what to do. So that really motivated me too. And so I went back to Kate and I was like, you're right. Let's do it.
Yeah, that resonates a lot. I mean, in my experience, people are so worried about the presence of pathogens that they do a lot to prevent them. And many of the things they do to prevent them are actually at the expense of mutualists who could in turn be actually a natural mechanism to prevent against pathogens. And like we see this for a long time in the South, they would fumigate the soils in the nursery with methyl bromide. Um,
Oh, wow.
to kill everything, right? Because they don't want root pathogens. And in that process, they're certainly extinguishing like populations of mycorrhizal fungi that could prevent against those root pathogens. And that
Sure.
happens to this day.
Interesting.
Um,
Interesting.
it's totally wild. We got a question from the chat. Interested in GK's results. They're very interesting and they want to know more about diversity, alpha and beta of soil fungi in the urban environment compared to the rural forest. I'd love to know that too.
Right. Okay. So here's a very interesting thing. I don't have the data on this slide deck, but what I will tell you is when there are ectos present in the soil, diversity goes way down. Um, so for example, diversity in urban forests here in New England, or in Massachusetts, just because there's so many oak trees and there's so many ectomycorrhizal fungi in soil, it looks like those soils have very, very low alpha diversity. But at forest edges, when the ectos go down, the pathogens tick up, everything else ticks up too. A lot of saprotrophs increase for both the fungi and the bacteria, which I find very interesting. And so there's something going on. I don't know if it's competition or the ectos are suppressing the pathogens or vice versa. I'm not sure, but that's very consistent. Now it's interesting in street trees. this we find that pattern below ground, where in street trees we have very, very low ectomycorrhizal abundances and high diversity of microbes, other microbes. But the leaves are a different story. The leaves, if you look at like Fisher diversity or like Simpsons, from the rural forest into the city of Boston, it's like this, right? It was almost impossible for us to detect fungi and bacteria on leaves in the city. Um, but the ones that are there are super dominant. So it's just a very homogenous community on leaves in the city. And like I said, they're all pathogens. So the only fungi we could detect on leaves are sooty molds and animal parasites. And the only bacteria we could detect were zoonotic pathogens of humans.
That's
Horrible.
awesome.
It's not awesome, it's awful. But
It's
it's
kind of
very
cool.
interesting. It's very interesting. We're trying to think of titles for this paper because it's scary stuff. Um, there's
That's
another,
fascinating that the foliar endophytes are missing as well as the mycorrhizal symbionts below ground.
yes, yes, they are.
We
Um,
got a question from our very own Anne Rejo. Unmute and send it, Anne.
Hey, thank you so much for coming and presenting. This is all really interesting to me. I live in Philly, and so I feel you on, the concrete jungle.
yeah.
And I guess I was wondering more about that. Have you seen any cities where trees are doing like okay or pretty well? And is there any kind of relationship with like Singapore or something? Anything?
Now, so what I haven't found is that um, there are some places where post-planting mortality rates are lower than others. So I think in Indianapolis, the mortality rates were around 10% in the first 5 years after planting, which is pretty good because here in Dorchester, they're about 58%. So it's highly variable. We're trying to figure out what it is related to. So far it looks like it's moisture related, but there's probably other stuff too. Um, and there's not a lot of data out there. People aren't really quantifying that in many cities, I would say.
Yeah, that makes sense. I think, at Funga, we try to find places where the trees are growing better than expected. And so I guess if I were on the task, that would be what I would be trying to do. But it looks like there just isn't
Oh,
it.
you know what's so interesting too? Maybe this is a cautionary note. I'm not sure how you're looking for tree health, but in the city, the trees die twice as quickly, but they grow 5 times faster. So trees in cities grow extremely fast. We don't know why. We think it's partly related to light. It's partly related to maybe really easily available nutrients that are around in the city. Like I thought, oh my God, when we were doing this work collecting samples for these urban studies, I was under the Mass Ave bridge and I saw this guy with a Ziploc and he was throwing Cheez-Its on the ground for the ducks. And I was like, oh my God, I forgot the Cheez-It effect. Because we were trying to control so many things to just look at our. We found like sausages in the street tree. I mean, all sorts of disgusting things I won't tell you about, but also just like there's a ton of nutrients. And the way that I think about these trees is that it's we're starting to wonder if it's almost like diabetes, how with diabetes, you get unintended weight gain from consuming easily available resources, and that leads to other types of disease. I think the same thing happens with these trees. And so, you know, fast growth and like big canopies, at least in cities, does not equal tree health. And it certainly doesn't mean that there are a lot of symbionts below ground.
That makes sense. Thank you.
Sure.
We have a question from Gabe in the chat asking more about your thoughts about nurse plants and soil conditioning. And they want to know if, inoculation from an alternate host plant could dilute pathogens while maintaining some symbiotic overlap. This kind of sounds like what you were doing with the different types of nurses you were testing in California.
Yeah, I think it's a great question. The nurse plant question is really open. Nurse plants typically, at least I've seen in the literature and what I've talked to restoration ecologists about, is they've really been applied more for arbuscular mycorrhizal plants. I'm not seeing a ton of this on ectomycorrhizal plants. And so there's a lot to learn. I think, yeah, like we're kind of interested in this idea of nurse plants helping through below ground stuff, but in like the microbial community. But in deserts, nurse plants are really well known to provide water, to surrounding plants and like shade, stuff like that. And I think we have to be thinking more broadly about nurses than just microbes. But I agree, like there's definitely alternate hosts. I mean, we tested like three different types of nurses. I mean, how many plant species are there out there? And how many plants coexist with these trees out in natural forests? Like there's so many. There's so many we could try. Honestly, it's just a matter of like funding and manpower to figure out the best combinations, I think. Hi, Zoe, what's up?
Hi. I was wondering if the nurse plant idea, if it ends up producing amazing results in the California plots and you wanted to apply it to the urban ecosystems, how would you do that with the tiny little tree pits that are there now?
You can't. Although people are doing really cool things in Europe with nurses, like for example, with very salt tolerant plants, because one of the big issues we have in the city is in the temperate zone, we dump a ton of salt on the roads in the winter to break up the ice. And a lot of trees, it totally stresses them out. And that's actually why they die. And so people in, I think in Switzerland, are planting halophiles all around, like these little tiny ones all around their trees to try to save them. And it's just such an interesting idea. But that's very, very new experimental. Like it's not being implemented as a management strategy yet. But I think we could get really creative and think about like, what are the really tough things for trees? Water, salt, heat, pollution. So like, what can we do to ease those stressors for trees in the city? Because like I said, I mean, cities are hot and dry and, all these things because we built them for us, like because our buildings are doing that, our sidewalks are doing that, and that's not going to change. But the idea of maybe making these small changes to the green spaces, I think it's worth a shot.
I love that idea. I think it kind of reminds me too, of Suzanne Simard has this narrative around mother trees, and they do this in lots of different systems though, like the idea like people are doing clear-cut forestry, and what if you leave a handful of retention trees on the landscape? Can that lifeboat microbial biodiversity? So when you're going in and planting all the new seedlings, that those nurse plants are already there.
Right.
I don't know how much has been published on it though.
Yeah, not that much. Not for the ectos, like I said. Yeah.
Yeah. Do we go ahead, Zoe?
I was also wondering, I was thinking, Jenny, about the channels, like the little tree boxes that you were working with that had channels in between the different tree pits.
Oh yeah.
Any of that seemed to be working or not?
We don't know yet. That's a great question. So this is an idea of using root paths. This is an idea that comes from Vanessa Harden, who's a design student at Harvard School of Design, where she wanted to connect trees using root paths in an effort to connect them microbially as well and see what the impact was. So that's another part of the transplant experiment. We have these treatments where we're connecting trees and we've inoculated them to see if that helps. I think it's very cool. I really like the idea of building a city below ground. Like if we can't make the above ground more conducive to plant growth, maybe we can make below ground in areas that humans aren't using, like the spaces underneath the sidewalks that are just fill right now. The only issue with that, and I think that might be getting to the question from Joey in the chat, is cost. Route paths are very expensive to implement, and it's destructive to like sidewalks, and like basically the city doesn't want to do it. They don't have the money, they don't have the manpower to do that without like serious data, and us like going to whatever governor's, office and holding up our papers and yelling about it and trying to get them to change their practices. It's going to be tricky.
That's
Jerry
usually
has a
how change
question.
happens.
Yeah, well, right, exactly. Right. Yeah. So we'll see. We'll see what the data say, if it's going to be worth it or not. So urbanization reduces symbiosis. Yeah. What's the core issue? So I guess, so this question is about what's the danger of reducing symbiosis, maybe driving loss, or, oh, okay. So there's a couple things. There's a lot of benefit to trees in cities. They don't store a lot of carbon on a per hectare basis because we just have too many buildings here, too many people living. But they help a lot with human health. So the trees are providing a lot of shade, so they have a pretty strong cooling effect, and that's really well documented. They do some things like stormwater management. They also provide habitat for pollinators and stuff, but they can take up particulate matter into their stomata. They actually can they can take a lot of particulate pollutants like aerosols out of the atmosphere. Certain species can. So potentially reducing human exposure to those. And there's pretty good documentation of that as well. The other thing that became very apparent during COVID was that these trees are making people happier. People publish these really interesting papers on like visitation rates to trees in cities, and like conservation areas or like forest reserves just outside the city. And of course it exploded with COVID because everyone was lonely and sad. And then they would like do these surveys of these people and learn how happy the trees made them. So it has all these like positive effects on people in cities, because cities are like gray and terrible places. The other thing is that, and we recognize this, and the science is quite clear. So that's why cities are investing so much money. They're investing like hundreds of thousands of dollars a year. Major cities in the United States, but of course this is happening worldwide. And like the trees are dying. And they just keep doing it again and again and again. So it's a humongous waste of money and manpower. And when you talk to the people who are doing this tree planting, they're pissed. They're super frustrated. Like, this is the mandate coming down from the state. They have to do this and they want to do it, but they know they're just going to have to replant again next year in the same place. So, does that answer your question, Joey? I hope at least partially.
Yes, thank you so much.
Sure.
I guess to close out, let's say you knew all the answers to these studies and actually you'd replicated them 100 times and you're like, all right, I know what to do. How do you do it?
Great question. I have a lot of ideas about that, but they're all kind of harebrained. So I'm really, what do you think, Colin? I mean, this is something that I would love to be able to say, cities, this is the protocol for planting a tree. And I would love for that protocol to include cities farming their own microbiomes for trees. So cultivating not just trees, but the whole tree system, the little halophiles, the mycorrhizal fungi, whatever it is that is going to help these trees out, for these to be like sustainable operations. And that will have to vary from place to place. I mean, we're starting in New England just because it's easy for us. We're right here, but it'll be very different in other parts of the world. And that's kind of the exciting part. So I definitely, what I want is a protocol for how they should be managing the whole tree system. I know that's kind of a non-answer, but I think that's probably what would work the best.
No, I like it. It's like native trees, native microflora. And like that tree nursery needs to be more than a tree nursery. It needs to be a tree and fungus and other micro nursery. That's sweet. I'm all about that. Awesome. Well, I think that's all our questions. Thanks everyone for
Great.
joining us. I'm so glad we caught Mia's class for a bit. And thanks for your time, Jenny. This was awesome.
Yeah, thanks, Colin, so much for inviting me. This was really fun. And, if people have ideas about like this or, they want to get involved or they want us to try a thing, please let me know. I mean, like I said, we're pretty early on, you can tell in this, and we're excited about it. So, thanks again.
Awesome.
All right, peace out. See you
Bye,
later.
everyone.
Bye.